This little video is on mitosis and the cell cycle. The overall goal with mitosis is to take a cell and at the end of the process create two new daughter cells. For this lesson we'll have the nuclear membrane in green like this and the DNA in purple like this. Our simplified cell contains one homologous pair of chromosomes. It's obviously a diploid cell, abbreviated by 2N. It contains only two chromosomes, so 2N is equal to 2. This is an example of a somatic cell, otherwise known as a body cell, like the cell on your nose. Mitosis creates identical cells which we might think of as clones because they are exactly like the parent cell. For an average cell, this process might take 1 to 2 years. But some cells, such as the ones in your brain, live as long as you do. All right, let's quickly review the cell cycle. In this diagram, we'll start with our one cell here, and we will go through a cell cycle of 1 to 2 years. And at the end of this process, we will end up with our two clones. Cell cycle begins with growth 1. It then moves into a short synthesis phase, followed by growth 2. These three phases together, growth 1, synthesis, and growth 2, are collectively known as interphase. Roughly 90% of a cell's life is spent in interphase, with the majority of it being in growth 1. After growth 2 come the actual phases of mitosis, starting with prophase, going into metaphase, anaphase, and telophase. So this is mitosis. And the last bit is cytokinesis. Okay, in growth 1, the cell is obviously growing. It's doing all the many jobs that cells do. Makes lots of different kinds of proteins. And it's basically just kind of hanging out, being a good old cell. In the synthesis phase, the cell is making a copy of its DNA. It needs a copy because if the original cell had two chromosomes, each of the daughter cells need to have a complete set of chromosomes. Now in animal cells, something else happens in the synthesis, which is that the centriole is copied. That's my fishing reel, which you'll see later pulls the chromosomes. Plant cells don't have centrioles. In the growth 2 phase, the cell is basically preparing for cell division, and there are specific proteins that are needed for this part, namely the spindle fibers. Spindle fibers. Those are a microtubule network that pulls the chromosomes. The 4 parts of mitosis are actually to divide up the DNA into 2 groups. We're not actually dividing the cell yet. And finally, in cytokinesis, the main idea is to divide up the cytoplasm. And this creates two cells. Let's take another look in more detail at growth 1, synthesis, and growth 2. These green circles will represent the cell. The purple represents the nuclear membrane. And sometimes in growth 2, the nuclear membrane starts this process of dissolving. So I'm going to show it with dashed lines. And we'll put our chromosomes in red. So we have one homologous pair. Remember, synthesis phase is when the DNA is copied. So now we're going to copy that chromosome, and it's going to be connected by the centromere. So that red dot is the centromere. And of course, we need to make a copy of the other chromosome. And let's say we're dealing with an animal cell, which has a centriole, and that centriole would be copied in S phase as well. So there are the centrioles. And in G2, we'd pretty much have the same arrangement with our chromosomes that have been copied and connected by the centromere, and our two centrioles. In G1, the DNA fragments are called chromatin. But in S phase and G2, when they're connected, they're referred to as sister chromatids. Sister chromatids. One other thing, the DNA will have to condense in order to be moved around, and so it will look a little bit thicker, somewhat like what I've drawn here. Starts to become visible with the aid of a light microscope. Now in this part we're going to discuss the 4 phases of mitosis. We have prophase followed by metaphase, anaphase, and then telophase. Again, the green will represent the outside of the cell. So I'll draw 4 cells here. In anaphase, it may start elongating. And in telophase, it probably is shaped like a peanut, at least in an animal cell. If the nuclear membrane hadn't dissolved before, it's definitely dissolving now, and it will completely disappear by the end of prophase. The sister chromatids have finished condensing and would be very visible with the aid of a light microscope. The centrioles are starting to migrate towards the poles. So the poles of the cell would be represented by the top and bottom in this picture. And coming out of the centriole would be these spindle fibers, which are like fishing line. They're going to actually be pulling the chromosomes. Spindle fibers start to grow. Just a few things to remember for prophase, that the sister chromatids condense. The centrioles are moving towards the poles. The spindle fibers are growing out of the centrioles, and the nuclear membrane finishes dissolving. Okay, the main thing that happens in metaphase is that the sister chromatids line up on the cell's equator. which is represented by this orange dashed line. So we will put one pair of sisters right here and the other pair of sisters right here. The two centrioles in animal cells have made it to poles, so we'll put one up at the top and the other at the bottom. And the protein spindle fibers have grown out of the centrioles and connected to each pair of sister chromatids at the centromere. And for metaphase, remember that the sister pairs got to the equator, the centrioles got to the poles, and the spindle fibers grew and connected to the centromeres. In anaphase, this is where the sister chromatids get pulled apart by the action of the spindle fibers. So at this point, the chromosomes are being pulled by the spindle fibers to the poles. So finally, we can actually discuss the term chromosome. When they're individual like this, they're called chromosomes. Okay, in telophase, the chromosomes have reached the poles and they're starting to stretch out again. Like that. Two new nuclear membranes reform around each of the homologous chromosomes. In animal cells, the cytoplasm starts to pinch in like this, and the spindle fiber network dissolves and the centrioles basically disappear. Now the cell is ready for division of the cytoplasm. If we had been discussing plant cells, which are kind of rectangular shaped and somewhat stiff because of the carbohydrate-based cell wall, instead of the cell pinching in like in the animal cell, there would be the formation of what's called a cell plate that would form during telophase. So this is the cell plate. Here we'll discuss cytokinesis, which is the actual division of the cytoplasm. Basically, the cell continues to pinch inwards, and over a rather short period of time, two new cells would form like that. Each, of course, with a nucleus. And the DNA is definitely all stretched out long and skinny and would be called chromatin. Chromatin. So at the end of cytokinesis, we have diploid cells, 2 in abbreviation. And from here, this process, we would go into another round of the cell cycle, beginning with G1. And that's about all. Thanks for listening.